Hey there, future medtech stars! Have you ever thought a sugar could be the reason life works the way it does? Well, today, you'll meet two VIPs of the cell world, ribose and deoxyribose. They're tiny, they're mighty, and believe it or not, they help run your DNA and RNA. Let's find out how one missing oxygen atom can change everything. Ready? Let the sugar showdown begin. Take a good look at these two molecular structures on your screen. On the left, we have ribose, and on the right, deoxyribose. What small differences can you spot between these two sugars? Look closely at that second carbon atom. And why do you think these tiny structural differences might actually matter? That's right. You might notice a subtle but crucial difference around the second carbon. And believe it or not, a tiny change in a sugar molecule can have a monumental impact, especially when it comes to something as vital as our genetic function. Today, we'll explore just how significant these small sugars are. So let's start with the basics. Have you ever heard of the term pentose sugar before? The word pent gives us a big clue. Pentose sugars are simply sugars that contain 5 carbon atoms. They are fundamental building blocks found in some of the most important molecules in our body, DNA and RNA. Speaking of DNA and RNA, can anyone tell what these two amazing molecules do in the body? What are their primary roles? Exactly. DNA, or deoxyribonucleic acid, is our body's master blueprint. It stores all of our genetic information. And RNA, or ribonucleic acid, acts as a crucial messenger, helping to carry and use that genetic information to build proteins and carry out various cellular functions. These molecules are incredibly important, and a big reason for their vital roles lie in these pentose sugars that help build and support their structure. Now, let's take a closer look at ribose, the sugar found in RNA. Observe this structural diagram of ribose. Can you identify how many carbon atoms are present in this molecule? As we discussed, ribose is indeed a pentose sugar, a 5-carbon monosaccharide. It forms a crucial part of the backbone of nucleic acids, specifically RNA. Notice the hydroxyl group. That's the OH group and the 2-carbon here. This little hydroxyl group is what makes ribose unique and helps distinguish it from deoxyribose, which we'll look at next. So, where exactly in the cell would we find ribose? That's right. Ribose is a key component of RNA. It also plays a role in other vital molecules like ATP, which is our cell's energy currency. In RNA, ribose molecules link up with phosphate groups and nitrogenous bases to form the long, single-stranded backbone of the RNA molecule. This structural role is absolutely essential for RNA to carry out its functions, like delivering genetic instruction from DNA and helping to build proteins. Without ribose, RNA simply cannot exist or perform its diverse roles. Now that we've explored ribose, let's shift our focus to its close relative, deoxyribose, the sugar found in DNA. Here we have a structural diagram of deoxyribose. How does it compare to the ribose molecule we just looked at? What's the most noticeable difference? You got it! Deoxyribose is also a 5-carbon sugar, but is a very important distinction. It lacks an oxygen atom in its 2-carbon. Instead of a hydroxyl OH group, it just has a hydrogen atom there. This is where the deoxy in deoxyribose comes from. It literally means deoxygenated at that specific position. So, knowing its name, where do you think deoxyribose is primarily used in the body? Exactly. Deoxyribose is a sugar found exclusively in DNA. You can see how it forms the backbone of that iconic double helix structure. Deoxyribose helps form the incredibly stable backbone of DNA. This stability is crucial because DNA's main job is to store and protect our genetic information for generations. That lack of the hydroxyl group on the 2-carbon in deoxyribose makes DNA significantly more stable and less reactive than RNA. This stability is vital for maintaining the integrity of our genetic code. Now that we've introduced both sugars, let's put them side by side and truly understand their key differences. Let's look at them together. Pay close attention to the 2 carbon in both sugars in this comparison table. Can you describe in your own words what is different about them? This seemingly small structural change, the presence of an OH group on ribose versus just a hydrogen on deoxyribose as 2 carbon, has profound effects on the chemical behavior and, ultimately, the biological role of the nucleic acid backbone. Based on what we just discussed about stability, which do you think is more stable, the sugar with the OH group or the one without? That's right, deoxyribose is more stable. Ribose is more reactive due to that extra oxygen atom and its associated hydroxyl group. This makes RNA inherently less stable, but importantly, more flexible. Ribose flexibility is perfectly suited for RNA's diverse and dynamic roles in protein synthesis, where it needs to move and fold into various structures. On the other hand, deoxyribose provides the necessary stability for DNA, ensuring our genetic blueprint remains intact and protected. We've covered a lot today. Let's do a quick recap. Now, what's the main difference between ribose and deoxyribose? And where is each sugar found? Which nucleic acid uses which sugar? Why is deoxyribose so important for DNA, and why is ribose essential for RNA? What do these structures allow them to do? Finally, and perhaps most importantly for you as aspiring medical technology students, why does understanding these pentose sugars matter in medical technology? Think about diagnostic tests, genetic analysis, or understanding diseases. Understanding these fundamental differences is crucial for so many areas in medical technology, from understanding genetic diseases to developing diagnostic tests that target DNA or RNA. Knowing the basic building blocks like these sugars is absolutely foundational. Before we wrap up, please get ready for a graded test. Here are your instructions and questions. For true or false, read each statement carefully and determine if it is true or false. Write true or false next to each statement. Multiple choice. Choose the best answer for each question and write a corresponding letter. Matching time. Match each term in column A and its correct description or associated molecule in column B. Write a letter of the correct answer next to the number. Finally, for replication task, create a short comic strip titled A Day in the Life of Ribose and Deoxyribose. Work in groups of 3 to 4 members, and your comic should have 6 to 7 panels that creatively show the roles of ribose and deoxyribose itself, their structure, where they are found, and how the differences affect their function. Keep your content scientifically accurate, but feel free to give the sugars fun personalities and dialogue to make the story engaging. This project will be submitted online. You may use digital drawing tools or take clear pictures of hand-drawn comics. Note, the use of artificial intelligence for generating any part of the comics content is strictly prohibited. Original creativity and understanding are expected in all submissions. Please be guided by the criteria in grading your output. And that's the sweet story of ribose and deoxyribose. One's flexible and fast, the other's strong and stable. But both are essential in keeping our cells running and our genes in check. Now it's your turn. Bring these sugars to life in your comic strip and show us how structure shapes function. Make it fun, make it smart, and make ribose and deoxyribose proud. Thank you and see you on the next one, sugars!